All right. Let's get started. Good evening, good morning, everyone. So it's really thanks for everyone joining us today on our Immuseum seminar during the weekend. Particular thanks to our speaker, Dr. Oliver Harrison from Seattle, joining us today to give us the lecture. So it's absolutely my pleasure to welcome our today's speaker, Dr. Oliver Harrison from Benaroya Research Institute from Seattle. So Oliver currently is assistant member or assistant professor in Center of Fundamental Immunology. So Oliver completed his undergrad study in UK in University of Bath, and then continued his PhD training in immunology in University of Oxford, and then particularly working in the intestinal cytokine IL-18. And then from 2014, Oliver decided to pursue his postdoc training in NIID and NIH with Yasmin, and then did some marvelous discovery in the mucosal immunology. And then from 2019, Oliver started his independent research program as assistant professor in Center of Fundamental Immunology in Benaroya Research Institute in Seattle. So Oliver's study actually mainly revolved around understanding the mechanisms controlling host microbiome interaction at burial tissues, primarily the skin and the gut. These two burial represent the major portals of pathogen exposure and are key anatomical sites for developmental of inflammatory disorders. And then Oliver's lab investigates the molecular machinery of how T cells and B cell responses to commensal bacterials are mounted and function. The main goal of his lab is to understand how these immune cells promote burial tissue integrity and then repair, and then to understand how this goes away during disease. So Oliver's lab actually mainly interested in a few different aspects of mucosal immunology, which including to understand how commensal-specific T cells influence tissue homeostasis. And then second is to understand the mechanisms regulating poised immunity in various sites. And the last but not least, to understand how commensal-specific B cells responses are mounted and regulated. With a really a great number of new discoveries, Oliver received numerous awards, which including when he was in NIH, fellow awards for research excellence and Bill Paul awards. And then when he actually moved to Seattle, he got NIID Direct Award and Science and Non-Star Microbiome Award as a grand prize winner. So although I was when Oliver in NIH, I was didn't get a chance to hear his talk, but today I think he's going to give us a few taste of his recent work on commensal-specific B cells. And with that, I really appreciate that, Oliver, to offer us the chance to give the lecture, and then looking forward to your talk. Please.
Hey, super. Thank you, John, for the great introduction. And thank you for everyone for joining. Good morning, good evening. And if you're in Europe and you're watching this, I'm sorry you're not sleeping properly. Okay, so I will tell you a little bit about the work that we've been doing in my lab, looking at the responses of B cells to commensal microbes. This is very much our first foray into the world of B cells. So I am keen to get any and all feedback. There we go. Okay, and so my lab's really interested, as many others are, in really trying to understand how the commensal microbiota, or the bacteria, viruses, and fungi that reside upon and within our tissues, influence our host physiology. And really a body of work from many investigators, now over many decades, has implicated the absence, presence, or dysbiosis of this community, which is the kind of skewing of the community composition in a number of aspects of host physiology. And so those include our metabolism, actually behavioral and neurological development, the efficacy of things so powerful as cancer immunotherapy or checkpoint blockade, and increasingly now how these microbes influence the efficacy of vaccination. And really, as immunologists, we're obviously very biased that this is all because the commensal microbiota actually really underlies and is essential for the education, the development and function of the immune system, both locally within barrier or mucosal tissues, but also really systemically across body sites. And part of the reason that we think this is important, and one of the aspects that we study in the lab, is the induction of these cognate or antigen-specific T and B cell responses to these microbes. So this is very much not us saying this is the only way that this communication between the microbes and our body occurs. Certainly metabolism and engagement of innate and other immune pathways is clearly important. But we're really interested in these cognate T cell and B cell responses because they're able to tune the function of local tissues, but also recirculate through blood and lymphatics to other tissue sites to really have this systemic effect of immunity. And so whilst it was mentioned that really my expertise has been in T cells, we were clearly ignoring and needed to add this very important arm of immunity, so humoral or B cell responses, to our studies. And so we came in with a very kind of rudimentary understanding of what the field has learned over 100 or more years, really trying to understand the players in B cell responses within the gut. And so we came at this looking at secretory IgA, this soluble dimeric antibody isotype that's been really implicated in immunity to pathogens and is increasingly understood of how it contributes to commensal microbes and really immune homeostasis. And so this molecule has been implicated in both the neutralization of toxins as well as elimination of pathogens. But really, in the context of microbiome science, we know that this is really important for microbial confinement or the maintenance of the microbiome away from the intestinal epithelial cell layer that lines the intestine. But more than that, actually, it's really important for maintaining the diversity of this microbiome and keeping a healthy microbiome that's very diverse and complex. And in the loss of this kind of axis of IgA, this is lost as well. By contrast, whilst a lot is known about IgA function, we're really only beginning to kind of scratch the surface of the contribution of other isotypes to gut immunity in the context of commensal microbes. And really very nice work has implicated IgG recently through the labs of Megan Koch and Melody Zheng as a key role for IgG isotypes in immune homeostasis, and particularly within this early life or neonatal window, where these are really tuning actually the response of other adaptive immune responses to the microbiome. And that's very much at odds about what we know and what has been kind of perceived of the role of IgG responses to commensal microbes through work in chronic inflammation. And so that really is driven by observations like that there are class-switched and IgG responses to commensal microbes in a number of chronic inflammatory disorders. So, for example, inflammatory bowel disease, and particularly Crohn's disease, patients present with serum antibodies raised to OMP-C or outer membrane protein C from E. coli. And that's actually in some instances used as a diagnostic biomarker where this tolerance to the commensal microbiota is believed to be lost. But actually outside of the gut, this is also true. And in the context of patients with HLA susceptibility or genetic susceptibility to type 1 diabetes, Jane Danska's group have shown that there's actually anti-commensal antibodies in pre-diabetic children. And this is also true that there are antibody responses to a distinct number of microorganisms in patients with rheumatoid arthritis. How they contribute to disease is really poorly understood. And that's not something we're going to get into today, but suffice to say, a dysregulated immune system in the context of chronic inflammation seems to be also targeting the commensal microbiota as well as our self-antigens. So when we came into the field, we were really interested in how people are assessing this immunity to the microbiota, and really came across two prevalent and really sophisticated approaches that people are using. And one is really built out of the labs of Jeff Gordon and Richard Flavell, and really relies, this is this IgA, and now newer versions using IgG, really relies on the fact that the endogenous microbiome within the gut is actually actively coated with IgA. And so you can use that to immunoprecipitate these immunogenic microbes and really pull down IgA-bound microbes, and then using 16S RNA sequencing, or now increasingly metagenomic sequencing, identify the community composition of those microbes, eliciting B cell and IgA responses in the gut. And that's really powerful, but what it kind of lacks for immunologists a couple of things, which is it doesn't really tell you much of the kinetics of that antibody response, and it doesn't really tell you much of the phenotype of the B cell that produced it, or where that B cell was. And so it's a great first step, and it's actually a relatively easy way to identify immune responses to these microbes. On the flip side, actually some very elegant work by many groups, including Andrew McPherson and Fred Alt and Gabrielle Victora, have really built the ability to take and sequence the antibody locus, or the B cell receptor locus, from mucosal B cells, reconstruct those antibodies, and then use them essentially to pan for immunogenic antigens or microbes. And so that allows you to really phenotype the B cell that you're studying, but it's antigen agnostic, and you just know that it's probably something in the microbiome, but that's not necessarily even the case. And some of these antibodies are obviously responding to other antigens, including food. So they're both very powerful techniques, but we wanted to try and kind of merge the strengths of both of them to really start being able to track commensal-specific B cells in vitro and in vivo. And to do that, we wanted to start by picking a microbe of interest that we could use that was also well described. And so when we did this, we really came into this with three, I think, very simple questions. We needed to find a microbe that had been previously described, just full stop. We knew that it was, and it could be isolated and studied. We, as people that were reasonably confident about our T cell expertise, but very much not confident about our B cell expertise, wanted to at least focus on something that was T cell dependent. And if possible, although this was much harder to do, find something that had been described to elicit both IgA and IgG responses. And because we were working in the small intestine, we were really interested in how these T cell dependent antibodies were made. And that's really within these Peyer's patch sites, if you can see my mouse, which are really major inductive sites within the small intestinal lamina propria wall. And essentially, they're able to directly sample the luminal contents or the gut microbiome directly, and here set up T cell dependent antibody responses. And so with all of these things in mind, we really focused on a single immunogenic microbe, a really prototypic commensal that's been very well described in the mouse immunology space, which is segmented filamentous bacteria. I will use SFB, as many people do, because I can't say that throughout the talk. And SFB is really a very interesting microbe. It's very immunogenic, engages the host immune system within the mucosa very well. And it does so by colonizing the small intestine, and particularly the ileum, which is the terminal section of the small intestine. And therein it induces antigen specific to its own antigens, essentially, Th17, as well as TFH, or T follicular helper cells, as well as IgA responses. And part of that immunogenic behavior, we think, is because it is really intimately associated with the intestinal epithelium, and that it's actually adherent to the epithelium. It can burrow through the mucus and really engage with the host itself. And a lot of these phenotypes and descriptions about this immunogenicity arose very nicely from work out of Dan Littman's lab, and the study of the immune response within two commercially available strains of black 6 mice. So those either available from here termed JAX, but Jackson Laboratories in the US, and then Taconic Farms or Taconic Biosciences. And here, these two pictures are essentially showing scanning electron micrographs of the villi of the small intestine. And you can see the heavy burden of segmented filamentous bacteria really lining the guts of these mice from Taconic farms that's absent in mice from Jackson laboratories. And so that's really important because that's a system that we're going to rely on throughout. Because whilst SFB is not inculturable, it's very difficult to culture. And so people use, as we do, the transplantation or fecal microbiome transplant from an SFB-bearing mouse into a naive mouse to elicit colonization and then track immunity upon that colonization. Okay. And so what I told you about IgA-seq and then VDJ-seq of B cells is that we didn't really have a way to be able to track antigen-specific cells. De novo, we could find them, but it was a very labor-intensive means of doing so. And so we wanted to use a system where we could identify immunogenic antigens and then track the cells that recognize them. And so to do that, we turned to a system called phage display or phage display library screening. And for those that aren't familiar with this, which actually we weren't at the time, it's a really high throughput way of looking for protein-protein binders or interactors, or actually protein and other macromolecules. And in this instance, we are essentially using serum antibodies or antibodies isolated from the intestinal lumen to pan for binders from the SFB proteome. And so to do that, we take SFB genomic DNA from mice that are monocolonized with SFB, fragment it and repair it, and then clone it into a phagemid library. And at this point, these inserts can go in any orientation. They may have stop codons. They may be in or out of frame. But they're packaged into this phagemid vector, which essentially is an essential component of a bacteria phage, which is a virus that infects a bacteria. And so this is our phagemid library, but there's been no selection on this at this point. And we package this into E. coli so we can propagate it and maintain it. But the really useful step is this genetic complementation step, wherein we can now infect these E. coli with this P3 deficient hyperphage. And this relies on genetic complementation to really produce infectious phage. These bacteriophage or phage are able to display SFB antigens fused into this surface P3 protein, essentially displaying it on the surface. But they can only do so if the phagemid library and the phage and the P3 fusion is in frame, hasn't got any stop codons, and is in the correct orientation. And so that now takes us from a kind of random sampling of proteins into an ORF, or open reading frame, orpheome. And so these may be random fragments of protein, but they're able to be presented on the surface. And now we can pan this library, in this case of about 50 million independent clones, for binding to IgA or IgG from colonized mice. And so we do that essentially by immobilizing, here it says serum IgA, but this also works again for luminal IgA, down onto a plate, and then over this, passing this library of SFB-bearing phage. And here, over multiple rounds of biopanning, we can actually enrich the phage that we think are bearing SFB antigens. And in a process where we do this 4 times, we can actually be increasingly stringent in our wash and salt steps to really get high affinity binders from the SFB proteome or orpheome that are bound by IgA or IgG. From there, we can elute those and then generate individual monoclonal phage and retest those for binding by either IgA or IgG from the serum. And from there, our positive hits, we take the phage phagemid back out and isolate it for Sanger sequencing. And that allows us to link the genetic information underlying that antigen back to the protein that's being recognized. So what this looks like when you do this, and you, on the left, do our monoclonal phage ELISA, is that we can identify very clearly antigens that are bound by either IgA or IgG, and in some instances, both, with a clear background present in the germ-free, and some antigens actually in the library that are not bound. And a number of these are actually derived from mouse genomic DNA. So because we isolate the poop of these mice, actually some of the intestinal epithelial cells and the genome come with it. And so actually the contamination that's not being bound is often mouse. What I'll tell you is when we take in each of these individual monoclonal phage hits and we align it to the SFB genome, in red, you can actually see now not only within these described proteins, but really the epitopes within these proteins that are bound by these serum antibodies. And so the one I'm going to draw your attention to and the focus of today's talk is really this protein SFB3340. And actually that it's being targeted in different epitopes by both serum IgA and IgG, as well as luminal or gut IgA. Now, whether that's important, we don't know. But what we can tell is that actually this N-terminal fragment of this protein seems to be targeted multiple times in multiple mice, so is really quite immunogenic. So we got really interested in SFB3340 for other reasons as well, and that it's also actually been previously described as a very strong T-cell antigen within the gut by the group of Dan Littman. And they've actually built T-cell receptor transgenic mice that recognize a peptide derived from this SFB3340 protein in the context of IAFB, or mouse MHC2. So we really chose to study this, not only because it's already been identified, but tools have been generated to track the immune response, or the T-cell immune response to this protein. And so furthermore, the means by which this antigen is sampled by the immune system has actually also been partially described. And so in the left in paper from in work really using scanning electron microscopy, you can see that these SFBs actually burrow and are very adherent into the epithelium within the small intestine, and they actually disrupt the actin architecture of intestinal epithelial cells as they do this. Furthermore, in even better electron microscopy, when you stain for this protein, you can actually see there's an SFB antigen P3340 is present within the cytoplasm of intestinal epithelial cells. So this has been actively taken up or endocytosed into these epithelial cells. And so really that's probably one of the reasons why it's so immunogenic to the immune system. How this T cell and B cell response is mounted from this is still being worked on. But so what I'm going to tell you today is really focused about this protein and how it engages the immune system, and particularly the B cell response. So we found this antigen, and we wanted to make sure that actually it is indeed a target of B cells. And to do this, we simply took serum and did serum antibodies using serum from Jackson mice that do not have SFB or taconic mice that do. And what you can show is very nice serum titers of both IgA as well as IgG1 and IgG2b against this antigen, only in mice from taconic farms that are SFB colonized. And actually, if there are any isotypes that are not shown here, we have tested them, but they don't seem to make any response. It's just these three isotypes. What we can also do is use this antigen to stimulate B cells isolated from different tissues. And so in the case of these taconic mice, we can isolate and stimulate B cells from both the bone marrow, or including the bone marrow, the mesenteric lymph node, Peyer's patches, the spleen, as well as the lamina propria itself, when those are most likely plasma cells. And so that's great. It allows you to do a lot of things now knowing a B cell antigen, but it doesn't really allow you to profile the cells in great detail. And that's actually where we turn to using an approach used by the field of B cell immunologists, but widely used looking at pathogens. And so that's the use of B cell tetramers, which I always kind of think is a fancy way of saying fluorescent protein. But essentially what these reagents are is a streptavidin PE phycoerythrin core around which we've tetramerized our biotin-lated antigen of interest. And so essentially this allows you to stain antigen-specific cells within a polyclonal repertoire present in wild-type mice and allow you to identify them by staining for PE. What's really important, and almost if not as important, more important actually, is the parallel use of another reagent, and this has been well described by Justin Taylor and others. This is the use of a decoy tetramer. And so within the naive repertoire of, say, a Black 6 mouse, there are many cells that will bind structural components of the tetramer, including his tags, biotin, and particularly PE itself. And so you need to generate a decoy reagent that has all of those components, but not the antigen, and then essentially redshift the dye to be able to tell true antigen-specific cells away from those that are just binding structural components of the tetramer. Basically, from there on in, you can use it as a flow reagent, so it allows you to detect these things by flow cytometry. You can, in instances, use that as a basis for magnetic enrichment. But actually, what I'm going to show you today from the payers patch is completely unenriched. So this is just the frequencies as they are present in the payers patch. And that's because basically those cells are not super amenable to being magnetically enriched and going over a column. And so when we built these tetramers with help from Justin Taylor, who was previously at Fred Hutch and is now at the University of Virginia, as well as Marion Pepper in the University of Washington's Immunology Department, they seem to work really well straight out of the gate. And so what we can see here, and these two flow plots are gated on total B and plasma cells within the Peyer's patches of mice, either from Jackson, SFB negative, or Taconic Farms, SFB positive. You see a small population of cells within the JAX mice, and this is significantly increased in the mice colonized with SFB, both in frequency and in number. When we actually go in and phenotype these further, what you can see is that those very few cells that are present in JAX mice are actually phenotypically naive, and that is that they're expressing high levels of IgD and IgM. And so these are really actually the cells that are recirculating and a part of the naive immune repertoire that could respond to SFB were it to colonize, but are actually still naive. But in mice that are colonized with SFB, these SFB3/340 specific cells are now significantly activated and engaged in immunity. That is that they are switched or class switched. They've downregulated their surface receptor expression of IgD and IgM, and are now expressing other isotypes. And then that's at a very much higher frequency than the polyclonal population of non-SFB3/340 specific cells within the same Peyer's patch of the Taconic mice. So these switched Ig or class switched commensal-specific B cells are actually uniquely or pretty much uniformly engaged within a germinal center response within the Peyer's patch. And that's identified here using staining for FAS, but also GL7 and the negative expression of CD38. And that's really striking in most instances, but actually in the gut, this is really well described. This is a site of constitutively active germinal centers responding to the gut microbiota. And those germinal centers are really this kind of training ground for this Darwinian evolution of the acquisition of mutations within the B cell receptor genes that contribute to the affinity maturation or the increased binding of antibodies over time. And so basically all of this in response in the gut of a mouse under homeostasis is really embroiled in this selective procedure. What that germinal center response is doing actually to commensals is still up for debate. Some people have described that actually broadens reactivity to different microbes, whereas others have described that actually this increases binding to particular antigens. And so this tool will now actually allow us to address that question. That's something I'll describe a little later in the talk. And so what this now allows us to do to phenotype these B cells ex vivo is actually to start to ask questions about anatomical location of these responses. And so what I've shown you so far is really the pooling of all Peyer's patches taken from along the length of the small intestinal tract. Oh, sorry, before I get to that, yes, I should mention, SFB is sufficient to elicit this response. So if we introduce SFB into germ-free animals, this B cell response doesn't need any other commensals present. And as you would expect for this germinal center response, this is entirely dependent upon T and T follicular helper cells. Okay. So I was beginning to start to say, looking at anatomical compartmentalization of this response, we were really struck by the work of Simon Milling at the University of Glasgow, and then of Daria Esterhazy and Daniel Masida of Rockefeller and the University of Chicago now, really describing the role for different lymph nodes within the mesenteric lymph node chain that drains the gut and distinct segments of the gut as being able to elicit different T-cell effector responses. And so we asked, is there a similar situation within the Peyer's patches of the intestine along the length that goes from the duodenum or the proximal region through the jejunum, the middle, and down to the ileum in the terminal region? And now we can basically isolate Peyer's patches from those individual sites. And what you can see is that when we're looking at these SFB-specific cells, actually they're embroiled in germinal centers nearly to 100% all along the length of the intestine. But their effector function or their production of distinct antibodies actually changes by site. While we see very high serum titers of IgG1, we actually see very low class switching to IgG1 expression within these germinal centers, and that's true across the length of the gut. But actually, if we look in these proximal or duodenal Peyer's patches, we see very high switching to IgG2b at the expense of IgA, and that situation is flipped when we start looking in the terminal ileum or the distal part of the small intestine, where now we see very high switching to IgA, again at the expense of IgG2b. What is controlling that? Whether it's the level of antigen because SFB colonizes the ileum, if it's something elicited by that colonization, we do not know, but it's an active area of investigation in the lab and something that we're really interested in trying to understand beyond perhaps like the known candidates of maybe TGF-beta and retinoic acid. So that's within Peyer's patches, but we're also interested to assess the difference between Peyer's patches and again the mesenteric lymph node. And now these flow plots are actually comparing all Peyer's patches grouped together, just for cell number essentially, compared to all Peyer's patches grouped together. What you can see is actually the majority of this response happens in the major inductive site, which is the Peyer's patch, and very low engagement of this response within the mesenteric lymph node. But numbers aside, the quality of this response has also changed. So in the Peyer's patch, we see high IgA with some IgG2b production, and this IgA is completely absent in the mesenteric lymph node. So whilst IgA can be produced in the mesenteric lymph node, it isn't being done so against this microbe or this particular antigen. And that kind of makes sense if you think about the gut and how the gut-associated lymphoid tissues are set up anatomically and structurally, which is that induction of IgA is very important to try and prevent translocation of microbes past the intestinal epithelium. But actually, if you're now sensing antigen within the mesenteric lymph node, clearly these antigens have been able to translocate or have been translocated. IgA is probably not that helpful anymore, and now engagement of class switch recombination to opsonizing antibodies of like IgG isotypes is going to be more important to stop that kind of systemic bacteremia and the further translocation of gut microbes into circulation. So this tells us, actually, we think there are differences in quality of the response by site and tissue, and that's something that we're further investigating. But we were also really interested by a recent paper from Joan Yuan's group, I think at Lund University, really detailing how early life B-cell responses actually seed very heavily into the IgA positive population of B-cells in later life in adults. And so now we were going to really interested to ask kind of some kinetic questions about how these responses are established. What I've shown you is in adults, but we were really interested in looking in the neonate. And so to do this, we set up very simple experiments of timed breedings between isolated JAX mice having SFB negative pups or taconic mice having SFB positive pups, and then just compared in the stool by qPCR the abundance of SFB over time. And what you can see here at 2, 3, and 4 weeks post-birth is that actually SFB isn't colonizing the stool by about 2 weeks. This really happens as the mice are weaned and transitioned off breast milk and onto solid food. And you can see in the taconic mice, in gray, this should be labeled in gray, that they're starting to be colonized. And that colonization actually hits the level of an adult mouse by about 4 weeks of age. That makes sense when we look at the B cell response and use our tetramer to track the activation of these germinal center responses in Peyer's patches. We don't really see a response within about 3 weeks, but about a week later following colonization, now we see this germinal center response kicking off in the Peyer's patches. What surprised us a little bit, and we had to go back and read some more, was that actually by 2 weeks of age, you can find serum antibody responses to the same protein for both IgG1 and IgG2b against SFB in the serum of mice born to TAC or SFB positive mothers. And so again, this is before colonization of the pup and before activation of the B cells. And actually, these responses are inherited maternally, both through systemic circulation and probably through breast milk. And actually, now we can see maternally derived antibodies in the circulation of the pups. That's not true for IgA because IgA is actually not reuptaken from breast milk into systemic circulation, though we suspect you'd probably find IgA within the gut and the lumen of the pups that's derived from mum. And so now that gives us tools to really address actually in early life and kind of tease apart maternally derived and offspring derived B cell responses to the microbiota without some of the very powerful but sometimes labor intensive systems of kind of cross fostering or B cell deficient mothers, which have their own inherent alterations in mucosal immune activation. So what I've shown you so far, just check on time, good, is really natural colonization, vertically transmitted from mother to pup or dam to offspring. But we also wanted to check how these responses can be elicited de novo to really investigate more of the kinetics and phenotype in that context. And so, as I previously mentioned, we can conduct FMT or fecal microbiome transplant from a mouse that does have SFB into one that doesn't. This works if you use a taconic mouse as the donor, but actually we've also tended more recently to using a donor that is deficient both for RAG as well as IL-2 receptor gamma. And the reason for that in these lymphocyte deficient mice that don't also have ILCs is the level of SFB within the population is increased. That enables a more kind of reproducible colonization. But actually what we think is also really important is that those SFBs that we gavage are not themselves endogenously coated with antibody because these mice have no IgA and no B cells, or no B cells and therefore no IgA. So this is really antibody naive SFB that we're introducing, and we can actually track this response over time. And so when we do this and we look in the Peyer's patches now of naive mice, we see very few, if any, activated B cells. But this response is really engaged by about 14 days of post-colonization. And is actually very stable over time after that. And so I'm only showing day 28 here, but actually months after colonization, we can still find this response ongoing. And that's because SFB is stably colonized. And this constitutively active germinal center continues to progress. You can see that here in the bottom, but actually when we assess the quality of this response by staining for antibody isotypes, you can see that actually this is also stable over time. We see very low induction of IgG1 in orange, a reasonable induction of IgG2b in purple, and then when we gate on those cells that are negative for both of those, they are all IgA expressors, and that is basically maintained through colonization. So phenotypically, we wanted to kind of investigate the makeup of this response in more detail. And to do so, we turn to single-cell RNA sequencing, because now we can pull out antigen-specific cells of interest by flow cytometry and sorting. So to do that, we colonized JAX mice, 10 of them with SFB. And at day 21, when this germinal center is really starting to get going, we isolated all Peyer's patches from individual mice and then stained individual mice with oligo-hashtagged antibodies as well as the tetramer. We then pooled all of those samples together and just sorted a single population of SFB-specific B cells and loaded those onto a 10x genomics chip. And the reason that we hashtagged is that we can post hoc deconvolute both the transcriptome of individual mice, but also the B cell receptor repertoire. So we essentially get 10 goes at assessing the BCR response to this microbe. And when I show you some, I think, fairly uninformative UMAP plots, you can see that actually these phenotypically and transcriptomically kind of mirror what we see by cytometry, which is we have a nice population of naive cells, a germinal center that can be broken down into fragments of light zone and dark zone that we won't get into today, as well as a small population of antibody secreting cells that still maintain enough B cell receptor expression or surface BCR expression to be able to bind the tetramer. This is kind of reflected in the isotypes that these cells produce. And really, this is just to say that this really, our sequencing kind of phenocopies our cytometric analysis. I'm not really going to say anything more about the transcriptome. Hopefully, in a couple of years' time, Chuan will invite me back and we can tell you more about what's transcriptionally unique and why you should care about these cells. But what I'm going to touch on very briefly is really the B cell receptor evolution and utilization of these SFB-specific cells. And so what this UMAP is showing is actually projected using those oligo hashtags to deconvolute individual mice. And so each cluster is essentially an individual mice, and the light blue cells are showing you cells that have a productive sequence for the B cell receptor, both heavy and light chain. And then the dark dots are examples where the same usage of the V, D, and J gene for the heavy chain and the V and J genes for the light chain are used to bind this tetramer across different mice. And so essentially all of these mice are using the same combination of B cell receptors to respond to this antigen. And that's what we're going to refer to as public clonotypes or recurrent clonotypes. And so those are what we're going to focus on because we have a lot of statistics to essentially address these. And what we can do is really take those recurrent or public clonotypes and do phylogenetic reconstruction of the evolution of these from the unmutated ancestor or the germline variant of these antibodies, and how as they're going through rounds of the germinal center, editing and then testing that B cell receptor, they're acquiring somatic hypermutations. And what we're doing is here for one clone, VH153 RC, and then from a couple of mice for a different one, VH176, picking different nodes from this evolutionary tree, generating monoclonal antibodies from those sequences, and then testing them for affinity against SFB3340 protein. And what you can see, whether that's by ELISA on the left or by biolayer interferometry, which I can only say once, I'm afraid, so BLI, you can see that as these cells pick up mutations, somatic hypermutations, that's actually actively contributing to increased affinity maturation. So this is really the first demonstration with a known antigen that actually a somatic hypermutation in the gut is driving affinity maturation to commensal antigens under homeostatic conditions. How that maintains over the 6 months of colonization and as long as I'm willing to pay mice, we don't know. But we're really interested in getting into understanding how the dynamics of those germinal centers and how new naive cells may be feeding in and undergoing this process. What I can tell you is that when we make variants of these monoclonal antibodies, based on the unmutated ancestor, so none of the essentially mutations are there, but we just introduce single mutations into the CDR3 region of these antibody heavy chains. Actually, even single mutations in the CDR3 are sufficient to drive this increased affinity, in line with what we know about CDR3 mutations driving affinity in response to vaccines and pathogens. So finally, we want to take these antibodies and actually show that they're binding to what they're meant to bind to in situ. And so here you can see, which is a confocal microscope image of the small intestine and particularly the terminal ileum of a mouse colonized with SFB, stained with these affinity matured antibodies that we generated. And you can see binding of puncta around really the tip of the intestinal villi where SFB is known to reside. So we think that these monoclonals are also able to bind SFB3340 in situ, kind of verifying that they are specific to what we think they are. Okay, so to surmise what I've shown you today and the work that we've been doing and where we want to go with it, we've really kind of built some of the tools to be able to start tracking commensal-specific B-cell responses in vivo. And we've done that by focusing on a prototypic commensal SFB. using phage display as a means by which to identify immunogenic antigens, and then using those antigens to generate B cell tetramers that now enable us to identify these cells ex vivo. And through cytometric and sequencing approaches, we've demonstrated really that there is an anatomical compartmentalization of these responses along the length of the small intestine that we're still trying to investigate, as well as between the Peyer's patches and the mesenteric lymph node. That within the Peyer's patches, these cells are undergoing constitutively active germinal centers that are picking up mutations that are contributing to affinity maturation. And where we're trying to go with this now is really to extend this to more microbes of interest, both bacterial, but also increasingly non-bacterial components of the microbiome. One of the joys of B-cell tetramers is that you're not confounded by MHC restriction. And so actually this now allows us to look at the antibody response to different microbes across not only mouse strains, but also species. And so now we're able to go in and ask questions about immunity to microbes in humans as well. Furthermore, we've set the stage really describing how these responses occur under homeostasis, but we're really interested in what happens when you start perturbing the GI environment, be that in the context of malnutrition or infection or inflammation. And so that work is ongoing. And so I guess the most important slide is really to thank the people that have helped us and helped Dr. Sheenan Verma, who's a postdoc in my lab that's driven this right from the get-go, as well as other members of my lab and help within Seattle, both at the Benaroya and other institutes, including the University of Washington and Fred Hutch. And then we have some fun ongoing collaborations, and we've got reagents from the other folks that you've seen on the screen. We have been funded by the other folks, including the NIH. And I am happy to take any questions if there are any. Thank you.